won't fall into a million pieces, but I've never collected this sponge before, so I'm just making a guess based on what it looks like its texture might be. But it looks soft.
Do you want me to push in more, or is this fine?
This is fine.
Okay, cool.
What's the finest ID we had on this? Did we?
I was thinking it looks kind of like a mycalae. It's definitely a dema sponge.
Yeah, for sure.
But you could put down mycalae maybe. Of course, it could be something completely different that I've never heard of before.
Yeah, also interested. Chris has a guess that he's willing to share.
Oh yeah, it's definitely squishy. That was oddly satisfying.
Nice. All right, it's up to you, Jake.
Yeah.
The two little
We
back
put it in a
boxes.
little back box, yeah.
Okay, cool.
All right, go ahead and hit the sample cams. All right, open the box.
So,
Oh no.
Lost a piece of it.
Oh, is it gonna fall in?
you still have quite a bit in your claw, so that's good.
I totally thought it was gonna fall straight in the bin.
Wow.
Nicely done.
Jake is good. Well done.
Nice tipping.
Nice. Do you want to put the rest in the same one?
Yeah.
Yeah, great, thanks.
Bump out the box. Is that fully bumped out? Yeah. All right. Something right there. Oh. Lost a piece of it again. Not gonna save that one.
That's okay. I think
That
a
one's
couple
going
of
right
good
to
pieces got in there, so.
That's wonderful. Thanks, Jake. And before we move on, it'd be great to just get a tight zoom on the texture on the rock there.
Okay. All right, feel free to push in.
Yeah, a lot of stoloniferous, right? Oh,
No, those are zoanthids.
oh,
Yeah, see like the texture in the skin, it's like kind of grainy.
yep,
That's
yep.
typical of a zoanthid.
Great, yeah, that's great. Thanks. Thanks, Jake.
Yep.
Just restarted the camera too.
Got it.
Okay.
Big Chrysogorgia. Yeah, some massive sponges here.
Yeah, these are also demasponges called Poecilastra. They always grow in this sort of plate-like formation. These are really large ones, though. Normally, they're more like the ones off to the left, the
Oh,
sort of
nice.
shorter plates, but
Gooseneck barnacles there.
oh yeah,
Those are really,
they're very necky.
yeah. Yeah, maybe if we can get just a quick
Yeah.
snap zoom on
Quick
one
snap
of
zoom
these.
there.
Wow.
Very cool. Thank you.
All right, come on.
Nice. It's the first time we've seen something like that, right?
Yeah, we saw some isolated ones earlier on a dead sponge, but
Not that cluster of
yeah,
big
not
ones.
like this.
Yeah.
Oops, fish.
That is a cutthroat eel.
Beautiful large Chrysogorgia. I also keep seeing a lot of these bright yellow unstocked crinoids, mycalae tometra. Thanks, Chris. Another one of these primnoids, so we've also seen quite a few of them, like the Narella. So, yeah, Quasegorgid seem to be as another one of those potential Pleurocorallium.
Okay.
There's just a lot to see here. So many things.
Yeah, just as a time check, we have just about 8 hours of bottom time remaining. Actually, 9 hours of bottom time remaining, so we'll be up on deck at midnight tonight. Looks like we have covered about, yeah, we're getting about 1/4 of the transect that we want to set up to do.
I've been thinking that it might be good to collect one of these yellow corals.
Yeah, I think so. They are quite abundant as well. And
Nice.
so I think yeah, if whenever we're set up to do so, we should do that.
Okay.
Yeah, and just maybe one of the branches towards one of the distal branches.
Yeah, I'd like to get a zoom at the polyps, because I've been seeing a few different kinds. We definitely have the Acanthogorgia, but then we also have some other genera around in here.
All right, go for zoom.
So one way to tell Acanthogorgia from some of the other genera is that they can't fully retract their polyps, and they tend to have a little crown of spines. So this might actually be an Acanthogorgia. If Asako is watching, I know she'll probably have some ideas about this coral. This wasn't the one I had in mind for collection, but the polyps are a lot more dense here, so I'm thinking Acanthogorgia and not one of the other yellow ones.
Okay.
Like the one that had the snake snart on it. I was really interested in that one since I wasn't sure where to place that one. There is something on it though, if you can pan up just a little bit. I'm not sure if it's on the coral, if it's in the background. Oh, I think, oh, it's a sponge. Okay. What do you think, Daniel?
Yeah, I mean, unless there's any strong feeling here, maybe I'll see in the chat if Chris or others. I don't see any strong.
Okay, well, we can leave this one.
Yeah.
It's
Yeah.
not the one I thought it was going to be.
Okay.
Okay.
Oh, we can combine and continue on.
Thanks.
All right. Yeah. Well, we'll continue on and look for the others. And
So there's a question. What major characteristics do you look for species identification when you're far away?
Oh, can you repeat that?
Yes. Let me pull it back up. What major characteristics do you look for at species identification from far away?
Oh, well, it depends on what I'm looking at. So, when you're really far away, sometimes it can be hard to tell
fish
if
there.
something is, say, like a sponge or a coral. So, it's a lot of pattern recognition. I've seen a lot of these animals before, and so I remember kind of like their shape, their vibe. Once I can narrow it down as to what I think the animal is, like what phylum is it in, I'm looking for things specific to that group. Shape is a big one. Coloration can be indicative of some species, but then when we zoom in on, say, a fish, we're looking for very, very specific characteristics,
Can we
like
quick look on
does
that
it have
center
scales on the
screen?
top of
On
its
the
nose
fish?
or not?
Oh, in there, okay.
Or,
Well, actually, do the fish first, because
yeah.
he'll get
So
away
this
from
is
it.
a little, this might be a good example
Push in there.
of what I'm talking about. So we're looking at this little eel, and I know that it's in the family Synephobranchidae based on the shape of its head and the pectoral fins. As we get closer, I'm looking at its nose and the lateral line, and I'm trying to determine if it's in the Synephobranchus or Iliophis. So that one looked like a Synephobranchus to me because of the pointiness of the nose, the size of the pectoral fins, and it doesn't have the really distinctive white pores along the lateral line.
Jeff was also in particular interested in collecting if it's a small one.
The eel?
Yeah, he was interested in a small Synephobranchid, so I don't know if this is going to be feasible.
It could be slurpable.
It's a bit long,
And then
though.
I also
Is
like
it-
to
yeah, it is
close
long.
into this one, but yeah.
Oh yeah, that
maybe-
one's definitely got the sponge on it.
Yeah.
And then the other character with the Synephobranchids, the main one that we look for is the dorsal fin and anal fin positioning. The Synephobranchus have a dorsal fin positioning that is behind the anal fin origin, whereas the Iliophis, it'll be the opposite, where that dorsal fin starts before the anal fin origin. And how far back along the body that dorsal fin origin is on Synephobranchus will help us narrow down the species of that animal. Well, I think it went into the rock.
Okay, so you want to zoom on that?
And
So
this guy
yeah,
right there.
that
Yeah.
one,
All
that
right.
sponge, we think has the black coral in it.
Okay.
But say for sponges, sometimes coloration is important because live sponges tend to be this really bright white, whereas dead sponges can be more brown in color. We don't count dead things in our annotations, so I usually disregard those. The shape
I can push in there.
and the texture are also indicative. So the one we're looking in at, the sponge we're looking at now, is a farreid sponge that has this black coral growing on it. And this black coral is Antipathes xylospongia.
Yeah, that's going to be the easiest range expansion ever. Because, I actually had the privilege to describe the species several years ago with Dennis Opresco, and we described it at that time only from specimens found throughout the Hawaiian archipelago, and that's because those were the specimens that we had available. So this is a new record for Palau, but yeah, it's likely found throughout the Pacific in many places.
And the yellow coral to the left of it was the one I was thinking about for collection earlier. You see how the polyps can fully retract against the skeleton. And so when I'm looking at these yellow corals, that's what I'm kind of looking for. It's how the polyps act when they're closed can be indicative. This one has a more golden color than the other one we were looking at, which I thought was interesting and different from some of the other Acanthogorgiidae that I know.
Yeah, and several people here in the chat are agreeing if we're set up to do so is to try to collect a specimen of this yellow.
This one might be tough, but I can.
Yeah, it's kind of in this sort of scoop.
I can't tell how far away. There. Also want to make sure there's nothing above. Okay, I think I might be able to do it. Let's try it. See if I can reach. No, I don't think I'm able to reach. Oh. Yeah, that's as
No,
far
that's
as I
too
got.
far.
Too
All
far.
right. Well, we'll look for another one.
Yeah, Chris is in the chat here. This dive reminds me a lot of a dive we did out in the northwestern Hawaiian Islands on, I think it was off Necker Ridge or one of those really narrow ridges.
Yeah,
Similar community.
it does look similar.
Another one of those unbranched bamboo corals. Yeah, it's amazing how abundant that one sponge is that we collected earlier.
I'm a bit far away.
Yeah, I was trying to catch up, but
At the end of the leash.
Can we get a tight zoom on that from NOAA?
Sure.
Okay. So
Push in there.
yeah, that's great. I was also trying to, if we can pan up, there's a white kind of candelabrum shaped coral that's above us.
yeah, so that's a Paragorgia.
That's as far up as I got. I gotta.
Mm.
If you come a little bit wide.
Yeah, that one looks like it might be an Arella.
Yeah, this one is, I think Chris wanted to get a good close-up on that.
All right. Push in there.
Yeah, ideally I would like to take a look at the base, but it's hidden here, so I don't think we'll be able to do that. That's good. Thanks, Jake.
Okay.
So we'll continue moving here along this ridge, try to document things. I think we've mostly got our eyes out now for that yellow coral. We'll also, once we reach 1,000 meters, which is going to be a little further up, we'll try to get another water sample Is this the Megan?
It might be. If we can zoom it real quick, see if it is. It's really hard to tell from far away. The yellow all looks the same.
Go for a quick zoom.
Yeah, I don't think it is. It's too fluffy. Yeah, no, that's not the one. This is the Acanthogorgia.
Yeah, we saw this one earlier.
Wonderful, thanks. Just a question here from our viewers. Thanks for the questions being sent out through the website. And there's a question here of how do these deep sea Pacific species, how do their distribution in the Pacific, how do they overlap? Are they all, do they have wide depth, wide geographic distributions, or are they more endemic? And I think the short answer to that is that we don't know. So there's this phenomenon like when in the past there was basically every species that was described for one single location, and then at that point they were thought to be endemic. But over time, as we start to explore more throughout the deep sea, we start seeing similar species in different environments that were once thought to be endemic. And we'll maybe take another look quick at that.
Oh yeah.
Gurney asteroid sea star. I think that's the same we recorded limits to SAM. And so yeah, there's that's a question that will take a lot of time and a lot of observations to address somewhat accurately, when the vast majority of deep sea hasn't yet been explored. And it's difficult to say something about the distribution of a species, the true distribution. I mean, you know where certain things have been recorded, but there's just so many places we haven't looked.
So this cookie star is called Hippasteria. It's a family Goniasteridae. They are coral predators, so often see them feeding on different corals.
Do they have a preference?
They really like bamboos, but I have seen them feeding on other families of corals as well.
That sponge looks like a croc.
It does look like a croc.
Jake is always seeing shoes.
Yeah, that's an astute observation.
It's the important things.
Yeah.
See another one of those clusters of gooseneck barnacles. But we got a good zoom in earlier.
So a question from our viewers. They are asking if a lot of these animals are hard or soft underneath all the water pressure.
Well, it really depends on the animal. Like that one sponge we collected was definitely squishy, and the other sponge we collected was crunchy, like potato chips. So some of these animals are extremely soft, and some of them are very, very hard. Like crinoids, for example, they're very flexible and bendy. Some of the corals are very hard, and some of them are flexible as well. It depends on what you're looking at. They all live very different lives, and their bodies are specifically, adapted to the type of life they live. Now, if you're asking how I know if it's going to be crunchy or squishy, that's experience. And that experience comes from watching hours and hours of ROV dives and also working in the lab with specimens.
So a follow-up there, Megan, like what first sparked your interest in science and how long have you been doing this for?
So I first got into marine science when I was about 8 years old, and I learned that you could be a marine scientist, and I thought it was the coolest thing ever, and that's what I wanted to do, and I stuck with it. But my foray into deep sea ecology officially began when I went to grad school, and I was using video data from the Pisces submersibles to characterize deep sea coral communities on the Kona coast of the Big Island of Hawaii and try to understand better how these corals grow and how the communities form over time.
5 meters.
And that was back in 2013, so.
Yeah, red one top of screen maybe.
Wow.
I think it's in a bubblegum coral. And
Come wide on the Atalanta cam. I just want to look at the tether.
Yeah, the first dive I ever participated in was with the Deep Discoverer ROV. I was helping lead that dive from shore. They were on the Kona coast doing a transect for me for my thesis research, surveying the precious corals in that area. And then I was hooked. I wanted to do it again.
So a little bit over 10 years.
Mm-hmm.
That's really cool.
Yeah.
yeah, that's great. Thanks, Jake.
Alright. Come back on.
A viewer is asking, what do the crinoids use to adhere themselves to different structures or organisms?
Oh, so they have these little legs. So you have the swimming arms, those feeding arms that are, very feather-like. But then underneath them, they have these little legs called cirri that are prehensile, and they can grab onto things with them.
Yeah, if we can zoom in on the base of that guy.
Roger. Start pushing in there.
It's a Narella.
Yeah, Chris wanted to see some detail by the base, if this is Narella alata.
Mm-hmm. Yeah, sometimes it's hard to get to species with the Narellas. There are a lot of them. So Chris is looking at where the branching begins and how it is branching. There's also a small Enelopsamia sclerotinian coral down there near the base as well. We saw some amphipods along the branches.
All right, going wide. Yeah, there's a bit of a current.
Yeah, you can see some of the animals kind of moving and you're getting pushed around.
Yeah, it's just pushing the vehicle back, backwards.
Are you dead on into the current right now, Jake? Or
Yeah,
no?
it was just pushing the vehicle backwards.
So basically coming from the east to the west, which is, yeah, the oceanography. Yeah, if you look
It's
at the
just
broad
kind of
scale oceanography,
flowing down
that's the
the-
predominant
down
current flow. Right, basically have the equatorial current coming from the east to the west.
I bet if you look down that sponge, you'll see a shrimp.
You think?
Yep.
Megan, a follow-up question on your background. How much school and training did it take for you to get a job like this?
So I have a master's in marine science. So that was 4 years of undergraduate degree and then 3 years for my master's.
Oh, I don't see a shrimp.
Oh,
well, maybe.
there's got to be a shrimp in there.
Shrimpless and Palau.
I don't know.
I don't know. Maybe it's hiding.
It's a good bet. Good bet.
I feel like I see it was just underneath that curve of the sponge. They almost always have a shrimp inside.
I
Oh,
think that's the yellow is the
that-
one they wanted. All right.
Oh yeah, there's one further down, Jake, and there's one up there. I don't
Oh,
know
yeah.
if the lower one is easier.
Probably not.
No. These are both kind of challenging positions.
Long-legged shrimp. I haven't seen those guys.
Yeah, they've been around. They're just kind of chilling. Oh, yeah,
Yeah, it's gonna be
that's
difficult.
not gonna work. Yeah, different one then.
We'll keep our eyes peeled.
So Dan, a viewer wants to know if there's any plans, expedition plans for 2025 out yet?
Yeah, so we have, thanks for that question. And so we have announced on our website just our operating areas for 2025. So we know kind of the general areas that we're going to be working in and the sequence that we are. We don't yet have exact dates for each expedition. But we have a general plan, and in fact there is a call for science input out that allows anyone to submit priorities within the places that we're going to be working in 2025. And that's how typically on these NOAA-funded expeditions aboard EV Nautilus, how we plan. We basically ensure that these expeditions are not just addressing the intellectual curiosities of one or a few principal investigators, but really that they try to address the broader needs of the scientific and resource management community. So on our website, you'll find a link to our call for science input, and that outlines where we're going to be operating. And our general plan for 2025 is to, once we finish our season here in Palau in December, we'll be overwintering, and then starting in April or thereabouts, we'll do our shakedown expedition in Guam. We typically have a week-long cruise, and that's really just dedicated at making sure that all of our systems are ready for the field season, so that will be in Guam. And then we have 2 back-to-back expeditions in Guam and the Mariana Islands, one of which is going to be focusing on the Mariana Trench mostly, and then also using various profilers to sample both the bottom of the trench as well as the water column above it, as well as adjacent seamounts with our ROVs. And then we'll have a second expedition that is going to be focusing on mud volcanoes around the Mariana Trench Marine National Monument, work that's going to be in both Guam and the Northern Mariana Islands. So those are going to be our first 2 expeditions, and then after that we'll be transiting east and have an expedition in the Marshall Islands using our ROV to explore a wide variety of habitats around the Marshall Islands. That will likely include a whole bunch of biodiversity assessments of different seamounts and different environments, and we also have tentatively a couple of targets for underwater cultural heritage sites, and there's a few that have been lost there that are quite historically significant, including USS Pennsylvania. And then after Marshall Islands, we'll transit south to the Solomon Islands, and that will be an expedition focused exclusively on underwater cultural heritage sites. That's going to be using a mapping platform from our partners at the University of New Hampshire, the Uncrewed Surface Vehicle DRIX, that will map an area off the island of Guadalcanal, which was the stage of at least 5 major naval battles in World War II. And then we'll use those maps to plan dives using our remotely operated vehicles. And then after the Solomon Islands, we'll head east to Fiji. We'll be using both our hull-mounted mapping systems and our ROVs, much like we're using in this very expedition, to explore a wide variety of habitats in the exclusive economic zone of Fiji. That will include a diversity of different habitats, including some very interesting hydrothermal vent habitats. So the western portion of Fiji is known to include some pretty active hydrothermal vents, so we'll definitely spend some time in there, but also working in various other non-vent habitats, including things very similar to what you're seeing on screen here. So it'll be a very interesting, season for us. It'll be taking Nautilus into all places that Nautilus has never been before. So we are starting the process of engaging the resource management, government, academic, and other partners in these places to start planning these expeditions. There's very different ways you can get involved. I mentioned the call for science input is open and will remain open until January of 2025. They use that to develop our general expedition plans. And then we also have 2 other things on our website for those that want to get involved in these. So we have our Science and Engineering Internship application open. This is an opportunity for you to sail aboard EV Nautilus. It's a competitive process. We'll select a lucky number of interns that will sail aboard EV Nautilus. And then we have a similar program for our professional educators, our Science Communication Fellowship. These are also opportunities for educators to come sail on aboard EV Nautilus and become the communicators, become the storytellers of these expeditions. So I encourage listeners, especially from the geographies of where we'll be operating next year, to get involved. Now we want to make sure that these expeditions address the primary needs of the places where we're going to be operating and are co-designed with input from these places. And so it's a very exciting season to come. We're all looking forward to it. And then we're also particularly interested in connecting the past to the present to the future. There's a lot of insights that are going to be gained. Over the last several years, EV Nautilus and other programs have been working in and around the Central Pacific. And we've learned quite a bit. And as we peek further to the west, it'll be interesting to see how these ecosystems and places that we're looking at differ or are similar to what has been studied in the past. And so little by little, we're starting to get a better picture of what the deep sea looks like across this vast expanse of ocean, the Pacific, that covers 1/3 of our planet and is still this great unknown, but that connects humans across the globe and is in many ways the great emulsifier of these island places. And a lot of these communities have been connected for a very long time.
So a follow-up question about the expedition in the Solomon Islands. Will you only be looking at the Guadalcanal, or are the Rennell Islands and others in the west also areas of?
Yeah, so the primary focus of the expedition is on a narrow passage known as the Iron Bottom Sound. So this is basically an area that's about 20 miles wide and 40 miles across between the island of Guadalcanal, Salvo Island, and Nagala Islands, formerly known as the Florida Islands. This, as I mentioned, was the stage where several very significant naval battles occurred. We know from archival research that over 100 shipwrecks were lost in this area. Over 1,500 planes were lost in this area, and only a tiny fraction of those have been found. And so we'll have a 3-week expedition to try to map.
Oh, that coral has its polyps open.
Document. Oh yeah, that's great sight there.
Push in there.
Yeah, so this definitely cannot be Pleurocorallium. You see how it's moving? A Pleurocorallium wouldn't move. So it's got a softer skeleton. Could be a Paragorgia or a Subagorgia.
Yeah, that was a good find there, Megan.
It's nice to see them with their polyps extended. It gives you a better sense of what this animal looks like.
All right.
There's also some anemones as associates, and looks like an amphipod as well.
There's a crab.
It's a what?
A crab. It's walking towards the one we just looked at.
Oh.
Push in there.
What kind of crab are you?
Yeah, my Connie spotted a good eye.
Yeah. It looks like it might be related to the king crabs.
It's hiding.
Yeah, perhaps a paralomus. Yeah, it ran away.
What is that?
It's a jelly. It's seen better days. It's probably something like Paralia.
Oh yeah.
Oh, we like fish.
Oreo.
Oh, the Oreo fish.
Oh yeah. There it is.
It is.
Push in there.
Do these
So-
get bigger?
No, this is their max size, pretty much. The genus is Neocitus. And we call them Oreos because their family name starts with Oreo. So the family is Oreosomatidae. But no one wants to say that. That's too many letters. And Oreos are delicious, and these are not delicious, but they're very cute. Put its mouth out. That was great.
All right.
So it looks like we're getting close to halfway point between waypoints
Another fish.
2 and waypoint 3. So it looks like we've covered about 500 to 600 meters from when we started this dive.
Push in there.
All right, so if you listened to me before talking about the eels, remember we're looking at the dorsal
Scales.
fin.
Right.
Where does the dorsal fin start? Yeah, so it's behind the anal fin origin, which means it's in the genus Synaphobranchus.
Nice.
Other tells for this genus has bit larger pectoral fins than the iliophids, and then the jaw, the way the jaw lines up, has more of an overbite than an underbite.
Jeff says Breva dorsalis, possibly.
Yeah, it did look like it was really far back. So Breva dorsalis is the species that has the dorsal fin origin that's way, way far back on the body. And that one looked like it started quite far back.
What was that you just circled?
Yeah,
The
right
sea
here.
cucumber.
Oh, okay.
Let's get a good look.
Yep, push in there on the sea cucumber.
That one's got a nice texture to it. This is a sea cucumber in the family Synallactidae.
That's great. Thanks, Jake.
Yep.
And then there's a little anemone to their side. Could be in the family Actiniidae.
What, is that a shrimp?
Oh yeah, that's a shrimp, heterocarpus. If we zoom in, take a little better look at it. So these shrimps are definitely really interested in attending bait. They are scavengers. So we would see, you know, many, many, many of these coming to the baited drop cameras.
You can push in there.
And these are delicious, you know, FYI.
The shrimp?
The shrimp.
That's great. Thank you.
Okay.
Yeah, they had them at the farmer's market once, and I was like, well, obviously I need to eat them.
How did they collect them?
Deep traps,
Okay.
yeah. So they tend to be expensive because it's not as easy to catch them as some other species of shrimp, and they're not farmed.
Yeah, this is something that I learned when we were kind of doing our background research about what previous deep sea explorations had been done here in Palau, and there was some work in the 80s where they were set up a whole bunch of traps to look at, you know, what's there. Actually, yeah, that was to try to see if a commercial fishery could be established.
Yeah, and there were a lot of different species of red shrimps that were collected. And they're really hard to tell apart in video because we're looking at very small things on the shrimp, like how many spines does it have on its head?
Push in there.
This is a little rat tail fish or grenadier, probably Kumba. Because it's super cute.
All right.
The rat tails are also scavengers of the deep, and they attend the traps as well.
Another one of those aggregations of gooseneck barnacles.
Mm-hmm. They do seem to like grow in little clumps,
Yep.
which is interesting.
A question to our science team. What is the most interesting sample that we've collected so far?
Oh,
That's a hard one.
that's definitely a subjective question. I don't know. We've gotten several really cool looking sea cucumbers, and we have a pretty neat batfish. We were looking at it again last night. Jeff took it out. Yeah, I think for each person, they probably have a different favorite sample. Maybe for the Pickrick team, the water, the results from all of your water samples will be the most interesting. But maybe the water itself is not the most exciting to look at. I don't know.
I don't know. I mean, the water just looks like water.
Yeah, of course.
So we'll wait until we get the results. But I think the coolest thing that we've collected is the skate egg. I thought that was pretty neat, and I was looking at it in the lab, and it looked pretty cool. It's pretty big,
Yeah.
bigger than I thought it would be. So yeah.
Definitely. Yeah, it's hard to pick. We have so many nice specimens of corals and sea cucumbers, and this I really liked the woven glass spicules for the stalked sponge that we collected. Seeing that was really cool, and a couple of rocks. But yeah.
Can we take a quick
Oh
look?
yeah, that's a neat one.
Yeah.
So this is a hydrozoan, but it is its own like very coral-like structure. You zoom in on the base, you'll see that it kind of branches off from one another, kind of like a stoloniferous octocoral where they're kind of connected.
Is this the Solandaria?
It could be Solandaria, but there's one that lives a little bit shallower that is also a possibility.
Try to push in there.
Yeah, excellent. Looks like tuna things for this dive. Don't remember this particular anemone.
Yeah. Given its depth, Solandaria is probably the better guess.
Yeah.
But hydrodendron is also an option.
What's the white on the coral?
It looks like a snail. And then there is a squat lobster as well. Yeah, so like the Solandaria tend to be more solitary, whereas the hydrodendron do the sort of splitting thing where they grow in aggregations. Trying to remember how deep we might see the hydrodendron, so I'm checking the guide. So 500 meters is for that one.
Can we take a look at the top holdover? Yeah.
Yeah. Looks like a toppled sponge. Very old one.
Yeah.
No, he can push in there.
It's very weird. I'd like to see this one alive.
Yeah, it doesn't seem to match.
Doesn't match anything we're looking at.
Yeah, that we've been seeing in the background. That's why it caught my eye.
It could be like that Caliphacus that we saw with the multiple puffs. It had like 10 different heads on it.
Yeah.
That
But
that
it's
might
massive,
be.
yeah.
Yeah, this one's huge.
Wow.
So it'd be cool to see that alive.
You think that if it was a Caliphacus, those heads would have just kind of broken off, and
Yeah.
so then we would just be left with a sort of spine?
Yeah, that's usually what happens, is like the puff at the end is quite fragile, and there's actual cell tissue that's holding the spicules together, and when that dies, those spicules aren't knitted together as strongly as the stalk, so it won't stay together for as long.
Yeah, that's great. Thanks, Jake.
All right.
Nice. Chris Kelly says, too many branches for that multi-hued Caliphacus.
Yeah, but remember we saw like the one with 10 branches. So I know, Chris. We usually don't see that many heads, but then we saw one, and I was like, well, that's weird.
Maybe it's a special version.
Yeah, well, it could be
Only-
different species.
Special Palau Caliphacus.
Could be. You just never know. Every time we do dives like this, we're finding new things, especially in areas that we've never explored before, like Palau. These depths are all new to science right now. So there's been a lot of science done shallow, and there's been some science done down deep in the trench, but nothing at these depths in between. So this is really new and exciting first sights. And while you'll notice, oh, maybe it's this Pleura thing.
Wow.
So there are some Pleuras that branch and form fans, and then the mid-stalk would stay is firm and more compacted than the outside. I don't know. There's a lot of mystery down here. We can speculate, or in the case of sponges, speculate.
Unbelievable.
Oh, I can't take credit for that. That's all Chris Kelly. When he goes and does sponge work, he calls it speculating.
He's cracking up in the chat.
Well, he taught me well. Everything I know. Oh wow, this sponge is really cool. So this was a Pleura sponge, but it is thus passed on, but it's still a home for many, many animals.
Yeah, it'd be good to just get here the...
Yeah, push in there.
Oh, that
Yeah, we've
texture.
got a Brasingid sea star, hydrozoans, cladorized sponges. Looks like, see, there's another one of those little fuzzy things. It could be a cladorized as well. the stick-like ones. Yeah, oh yeah, it definitely is a cladorhizid. I love the zoom. Go Aaron on the zoom.
Yeah, it's really nice.
That's great.
Chris was saying that it settled on too small of a rock, so it probably just toppled over.
Yeah, that happens. It get too heavy. And it looks like all this stuff here is kind of loose. Oh, what was that? I don't know. It kind of looked like an ostracod. Very small crustacean. Oh, and look at this. I think this is the same type of crab as we were looking at before that went hid in the rocks before we could get a zoom on it. Looks like a small king crab, and possibly in the genus Paralomus.
All right, come on.
It looks comfortable.
Yeah.
Yeah,
Well, this
cozy.
seems like the nice place to be if you're a small crab.
That was really pretty.
Oh, and there's a new dema sponge. This one that's sort of encrusting could be something like Fecalia.
Barnacles.
Oh yeah, and they got some more gooseneck barnacles. Oh, there's a live Tretopleura, the live version of what we were just looking at that had toppled over. And there is a squat lobster on it. Might be a Pseudomunida. See a sort of a smooth body with very long arms.
What's our range to waypoint 3? How close are we?
We are about 75 meters away.
Nice. Be curious to see how this photogrammetry transect goes.
Yeah, I mean, there's so much to look at here.
Yeah,
Exactly.
I think in on that overhang is something that I don't think
Is this little yellow one down there, the one that...
No, that's a Anelopsamia, that yellow one. But,
Yeah, but
good
this,
spotting.
I think that is a new sponge.
That's a dead. The one that you circled is dead.
Is it dead? Okay.
Yeah, it's the furriid one, but dead.
Oh,
Oh.
never mind then.
Still keeping my eyes out for that yellow coral. Haven't seen one recently. There's a little Actinoscyphia, the Venus flytrap anemone. And so many sponges.
Yeah, there's quite the current now.
Yeah.
And you can see the tether being
Oh yeah,
pulled that
it's
way,
kind
and
of
then if I let go, I'm just drifting.
Well, we're almost at the top of the feature, which can cause current acceleration. And with how much stuff is here, I'm not surprised. These corals and sponges really like heavy current because the more water that flows past them, that more feeding opportunities they have. Ooh, that's an urchin.
Yeah, that's the first one.
It's really white.
Yeah, it's really pretty. We haven't seen something like that yet.
Yeah, first on this dive.
Is it dead?
No, it's alive.
Okay.
Push in there.
Yeah, no, they come in white.
And echinothruid.
Yeah, Spirosomanidae. Spirosomanidae. This is some family it belongs to.
Nice.
Nice.
It's a really pretty one.
Yeah.
I love their tube feet.
All right.
Some of the other echinothurids are really fun because at the ends of their lower spines, they have these little boots so they can walk across sediment more easily.
Yeah, maybe quick zoom on that guy too.
Okay.
And there's a fish right in front of it. Or no, oh sorry, no.
The
This
piece
is
of
toppled. Yeah, sorry. That'd be good too.
yeah, a piece of dead sponge floating down.
Yep. Yep, perfect. Thanks, Jake.
Yep.
Hemicorallium.
Would you like a zoom or is that?
Yeah, I mean it's if we.
Okay.
That's good. Another Farreid with the Antipathes, Stylospongia. But that one glass sponge that we collected is amazing how abundant that is.
I know. I've never seen that many of these kinds before, you know?
It's interesting how on previous dives as well, we were kind of seeing one sort of successful, if not species, sort of genus or family that is really abundant.
It's funny how that works, huh?
Yeah.
Or there's usually like,
Fish
it's like is it a sponge dive or a coral dive, you know?
Yeah.
So there's usually one that's
Holothurian dive,
Yeah.
black coral dive,
Yeah, maybe
sponge
on the
dive.
fish first and then on the sea star.
Okay. Can push in on the fish.
A rat tail? Is it?
All right. Come wide. I'll look at the sea star.
So Jeff is not quite sure.
Or did you want a better look there?
Yeah,
Okay.
at
I'm
the
just
fish. Before
Yeah.
he said this one was possibly a Kumba, but it could be something
Okay,
else.
push
so,
back in.
well, just Jeff is trying to think about potentially collecting this if we are set up to do so.
I can get out the suction.
Yeah, Jeff, if you please chime in here, but I think-
This is a very fast fish.
okay.
So if we try, you gotta
Gotta be quick.
do it fast.
Same, try.
Is that a yes or a?
I
Yeah,
think
let's
Jeff said let's do it.
Let's
Okay.
give it.
Come on.
I'm gonna just stop the camera.
Okay.
Yeah, I would
Roger.
turn the suction like all the way up and like just go for it.
Full jam.
Let's go to 4.
I'm gonna tilt down on the camera. Okay. You still have eyes on it? Goes down. Oh, I see it. Straight ahead. Up a bit. Oh, is that the same one?
Yeah, that's the one.
All right. Suction's all the way up. And it's on.
Oh. It kind of came at the sides, so.
Sediment against ground suction for a bit. I don't know if I don't think I can reach back there.
Yeah, it knows.
Yeah,
It was
it's
like, no, excuse me.
That's all right. Yeah, thanks for giving it a try.
Yep. Stuffed it into a rock.
Yeah, it was really fast.
Yeah.
It would have been sort of
Doesn't help that the current wants to just like
pull you
it.
around,
Yep.
yeah.
Push you right back. Okay.
We'll
Okay.
just restart at the camera here.
All right, do we want to keep following this feature, or do we want to head up on top of it to waypoint 3?
Yeah, I think we'll head in towards waypoint 3,
Okay.
because otherwise we're just gonna meander the entire seamount.
All right, that'll be 045.
And it is gonna start flattening out a little bit, but yeah, this. Yeah, we expected this narrow ridge to be this kind of the highest aggregations of animals, so I'm gonna make a prediction. I'll start dropping off a little bit as we go further in, and then I think we'll see another cluster towards the very top. Yeah, a lot of familiar faces now, so we'll try to zoom in on the things that we haven't seen before. And try to make some headway here. And it looks like we still have over 1.5 kilometers to go. We should have about 7 hours and 45 minutes of bottom time. And then looking ahead, tomorrow we'll have a very similar type of schedule as we had today, so deploying the ROV at 8 a.m. local time, and then recovering the vehicles at midnight.
Yeah, let's zoom in here and look at this red fuzzy stuff.
Okay. And just push in on the rock here.
This looks like Swiftia.
Yeah, exactly, and a lot of them.
Yeah, it's a lot.
That's great. Good eye. Keep building out our species list for this dive.
Mm-hmm. I think it's just going to get more and more diverse.
That's great.
All right, come on.
Oh, that's a pretty bathypathes.
That's also new for this.
Yeah, I don't think we've seen one of those today.
Yeah, and maybe if we can, like directly head on would be nice to get a zoom in on the middle.
Oh, to see if there's a worm?
Yeah, just a branching pattern also.
Yeah. Do you have an idea of the species?
Yeah, I think it's the pseudo-alternata we also recently described, but
We can push in there.
this. Nope, that's not it.
It's got a snake star, I think.
Yeah, that's.
Interesting. Are there ophiroids?
Yeah, yeah. And is there a way to just pan down a little bit towards the base?
Sure.
That's great. Thanks, Jake.
Yep.
I have a prediction that this video is going to lead to a highlight video.
It's definitely full of highlight options. I think we're good here, Jake.
Yeah,
Alright.
it's,
Should we turn off the lasers for a moment
yeah.
for highlight video's sake?
Scale lasers off. Okay.
Yeah, this dive has been quite extraordinary to the point that we're kind of becoming a little bit desensitized, and it's harder to impress us now. So let's see what happens here
Yeah,
as
we're
we
like,
keep
oh
climbing.
no, it's just another sponge.
Not me. I've been internally
That's
screaming
amazing.
for the past 3 hours.
That one.
Yeah, it looks like, is that another Swiftia on a hemicorallium?
Oh, no, it's a Novodynia.
Oh, yeah, yep. Sorry.
But that would have been weird.
Yeah, it just was very strange. Yep, no need to zoom in. But yeah, there's a lot of Swiftia at the base there,
Yeah, and
all around the
all
rock.
the little red stuff.
Yeah. Yeah, quite remarkable. And
Is that a sea star or not?
A sea star?
yeah, here we're on the eating on the
Oh,
pinoy.
yeah. I thought it was a sponge.
Yeah,
Good spot.
let's get a view of that.
Push in there.
Yeah, we do like to document these predation events. So this is a great example of how these cookie stars feed on corals. They're coral predators. They extend their stomachs out of their body and wrap it around the branch of the coral and then slowly digest it, which is really cool and super creepy. I know I said that they do seem to prefer bamboo corals, but there aren't that many bamboos to choose from around here, and they will feed on pretty much any octocoral.
All right. Come on.
We're currently doing a watch change.
There's a rock pen.
Hello.
Just give us a couple more minutes as we settle down the 4 to 8 watch.
And Jaden, just so you know, I was just told that we're slightly zoomed in on the Atalanta camera just to get a nice shot on Hercules, just so you don't think we're too close. So let me go full wide so you can get a sense. That's full wide there.
Nope. Negatory Ghost Rider.
So before we move, I have questions and I want to discuss where we move.
I have discussions too. We need to have a discussion, a group discussion. I'll let you go first since you're the watch lead.
I would be curious in seeing the eastern face of this feature at some point. Doesn't have to be right now, but I was thinking about instead of going straight up to waypoint 4, you know, kind of cutting across this low, this kind of saddle here at like, I don't know, 045, and then coming up a little on the eastern aspect of the next rise up towards waypoint 4. That was my thought.
Roger. Well, Chris, digest that.
Yeah.
Or do you got an immediate response?
No, we can do that as long as you're happy with it. I figure, so we could do something like, just kind of make a move like this, and then come up to like here and then climb to 4. That sounds good?
That's pretty much what I had in mind.
The steeper, the better. Is that what you're after there?
Not necessarily. I'm just curious. You know, the current seems to have gotten pretty strong up here from watching down below, and I'd be curious to see what the other side is doing in the current. Like, we're on the lee side, theoretically, of the ridge, and we're still got this community. I'd be curious to see if we see different organisms on the windward side.
Roger. I would be curious too.
What was your point of discussion, Dan?
My point of discussion, there has been much discussion. Jaden, right now it's selected as a destination, so any source, and it's ROV nav. I think. I don't know. Just press button, see what happens. Sorry. The lower camera currently is out of focus and has been.
For the entire cruise?
Yeah, pretty much. So, and also, we've not got a lot of quality still photos. So, that camera is maybe yes or no ready for the wild. You know, we're still working on it, but like right now, it's out of focus. So if we took a picture, we'd have a fuzzy still picture. I now have, we have control of it up here in the front row, so we can share the load a little if you want. One of the ways to get it to behave, besides beating on it with a big stick, is to cycle the autofocus. What I do not have up here is I don't have the knob to adjust the focus. All I can do is, oh wait, I got a button to. So if I take it out of autofocus, it should be reflecting what you're doing there, what I'm doing on your deck.
Yep, we're seeing the autofocus come in and out on our control panel.
Yeah, so as I go in and out, so I'm not a shutterbug, I'm not a photographer, I'm just a taxi driver, but I can tell you when, I can't see out the window very well. As far as operations, it's not a big deal because I'm using it more for, spatial awareness, but we need to get, as a group, working on still good quality still photos. So some of that is to get the shot framed up, right? And that's about all I can really do up here, especially when I have my hands full. And I can, as I have, take the still. And so one thing to consider as we're coming in on something that you might want a still picture of, I'm dragging a big dust cloud. So, you've seen it 100 times, Brian. I land and you get a really good look, and then all of a sudden, Hercules dust cloud obliterates what would have been a beautiful shot. So we're just gonna, I don't know, I don't have all the answers. I don't know what we can do, but we can, if we keep that in the forefront of our objective. So I have a personal stake in this. I have a bet with Jake Bonney on who can get the most quality stills out of a 4-hour watch. And there's a lot riding on it.
Well, my primary suggestion for that is somebody needs to fix the autofocus in order to make that a goal.
Roger. So if that's our take on it, then we can't get good stills?
Oh, we can certainly. No, I mean, I'm certainly in favor of trying to get, if we have a cinematic moment or something like that, we absolutely should take the time and do it. I am pushing back a little bit on it being a primary objective.
Right. No, maybe primary wasn't the wrong word, but just more, all I'm asking for is a little more focus on it. Ha, no pun intended.
Except pun, yeah, right. All right.
I can divert more of my attention to it.
Yeah, I can too. So it's going to be a team effort, and yes, it's not ideal yet, and the autofocus is frustrating. I want to, smash the controls up here at points, but yeah, I guess just play with it a little more and give us some feedback. What works, what doesn't, what's frustrating, what's nice, and it is a development project. That's it.
Okay. So before we get moving, can we take a look at this core right here?
Yeah.
I suspect I'm going to want to collect it. We've narrowed it down to 2 things, and we know it's one of those 2 things, but I don't think we're going to be able to for sure know what it is until we get it up to the surface.
Roger. I'm also going to, oh yeah, about the zoom on the lower one. If you're zoomed out, there's other stuff in the picture, like the porch and the manipulator at times. That mid-zoom is probably a good, and I'll try and land them like that, so they're in both cameras, which can be, it can be challenging, but, I'm going for it. Right now, sorry, I'm choking. More flying, less talking. One last thing too, the upper camera, when you're doing the timer photogrammetry, it should be as it is now too in mid zoom.
I don't even think we have control of the upper camera.
You do not, I do.
Okay.
But I'll zoom in and out depending on, like if we're working with a manipulator and then I forget to zoom it back in or something. So please help me out.
Copy that.
Remind me.
So Daniel asked us not to run the timer anymore on this watch until they go through what they collected on the last watch. So
Roger.
we'll just be in manual take mode.
Push the button as fast as you can. They're free.
I don't know if Mike would agree with that.
So far, I haven't killed anything pressing the button as fast as I can. Okay, Amber, you can zoom in for Brian there, please. Yeah, there's that dust cloud I'm talking about. Sorry, I choked. You need a closer zoom.
Nope, that's what we're looking at. This is either a white morph paragorgia or a Cypagorgia, and I'd like a clip of it so we can tell for sure.
Have we seen any like this in previous dives?
On previous dives, no. We've seen a lot of it on this dive. It's been a common coral this dive.
So I thought this was all rock, but it appears there's some.
This looks like coral rubble to me.
Sediment under it.
Yeah, and it looks like stony coral rubble to some extent too.
All right, I'm gonna have to get a little western with it here. I'm having trouble holding it. Why is that? Why? Can you give me maybe a little more delta? I don't know what's going on here. I can't find a good toe hold.
What species did you think it was again?
I think it's a white morph paragorgia.
Sorry about the viz, people. I'm just completely wrecked it. I'm gonna back off and try and come in from another angle. I don't really have a sense yet of where uphill is. No. That was a landing, a very unelegant one. Do you want a better, tighter zoom on it before we get it? Or
Nope, I got it last time when we looked at it.
So, what's going on there? This is when I'll typically zoom out on the upper so I can see the manipulator. And on the lower, you can push in there a bit, Amber. I can decide where I'm gonna ask Brian if he's alright with the snip.
Do you want any more than that?
Sure.
All right.
Is that red part also part of the coral?
What's that?
No,
Sorry?
no. We'll get it back. Sorry. You're fine, Dan. Keep going.
What? Can you describe where you want me to?
About halfway between the stalk and the base, you know, the typical 10 or 15 centimeters of it. Realize it's small, you may end up taking the whole thing. If it pops off, that's okay.
I can't tell what's exactly going on there.
So it's got, basically there's a main branch coming down here and going through there. And if you just want to like cut it right there, that's fine.
Okay.
And the whole thing may pop off in the manipulator.
Yeah. You might get some sponge and stuff with it. You all right with that?
Oh yeah.
Right there.
More,
here we go.
in some ways, more the merrier.
That rock's loose. You want the rock and the whole thing?
That's a big rock.
It is not. Look at the lower camera.
Let's get the coral and then we'll look at the rock.
Hey, it's a slurp shrimp.
Yeah, it's taunting you.
I like coral rocks, as you can tell. It's kind of cool when you pick them up. You get really good. But like you say, the rock smashes the thing in the firebox. I would call that an epic fail on the
All right, that works for us.
You good with that?
Yep.
This wasn't at a very good snippy angle. Remember, Harold, on the first day you were complimenting on my sampling, and I said, it takes 10 of those to make up for one of these.
I mean, this actually might be Pleurocorallium, which is not what I thought it was, but it was fragile. So what boxes are open?
Oh, we have the 2 big boxes on the starboard bio box that are free, and also C, the second to the aft, is available.
A little one, second to the aft. You don't want to burn the big one, do you?
If you can fit it in the little one, I'd prefer it there.
Yeah.
You are doing a better job than any of us could.